I will speak even more quickly than usual, so we have more time for Professor Rogers. I'll give a short intro. It's truly an honor to have Professor Rogers, John Rogers, to be the speaker today. He is an overwhelming research leader. I will tell you that his degrees were in chemistry and physics. His PhD was from MIT in physical chemistry. And in 1995, he joined, he was with Harvard until 1997. After that, he joined the legendary Biolaboratories. Then he spent 13 years with University of Illinois, Urbana-Champaign as a chair professor. In 2016, Northwestern stole him from U of I, and where he's a chair professor in material science and engineering, biomedical engineering and medicine, with affiliate appointments in mechanical engineering, electrical and computer engineering, and chemistry, where he's also director of the endowed Clary Simpson Institute for Bioelectronics. Professor Rogers has published over 750 papers, many in Science and Nature, and many I have read. He has been the inventor for more than 100 patents. And worth noting, not only he's academically beyond outstanding, he also has co-founded several successful technology companies. He has won too many awards I will not read. I encourage you to read online. But finally, Professor Rogers is one of the rare researchers who is a member of so many national academies: National Academy of Engineering, National Academy of Sciences, National Academy of Medicine, National Academy of Inventors, and the American Academy of Arts and Sciences. So without further ado, I'll give the floor to Professor Rogers, and I will give him a virtual round of applause on behalf of everybody. Professor Rogers, please go ahead.
All right, thank you very much for that introduction, and for the invitation also to come here and share with the broader community there some of the things we've been working on here in my group at Northwestern. I'm going to go ahead and share my screen and again get started here. So this talk is going to involve foundational concepts in mechanical engineering. I know there are a lot of mechanical engineers in the audience, but kind of at the intersection between mechanical, electrical, biomedical engineering, and medicine is kind of where a lot of our projects are residing, in terms of the landscape of interdisciplinary expertise and work. And so I'll focus on devices that we've developed to interface with the human body, specifically the skin, for a range of applications, all the way from physical fitness and sports performance to clinical medicine. I'll try to go fairly quickly in a description of the foundational engineering concepts that we've been using in this field over the last several years, almost a decade, I guess, by now. And focus instead sort of on the system level capabilities and what we're able to do, not only kind of in the the context of an academic laboratory, but in the real world, on actual patients and deployed kind of at scales that may allow us to have a positive impact on the way that we think about human health care in the future. And I'll split the talk kind of in two pieces. The first is going to focus on what we refer to as epidermal or skin-like electronics that embed microprocessor capability, memory, radio communication features, but arrays of biosensors where the skin is used as a window for quantifying various characteristics of underlying body processes to capture a clear physiological picture of health status. And then in the last piece, I'll describe devices that are similar to those epidermal electronic platforms in their mechanics and their skin interface, but differentiated from the standpoint that their functionality derives from lab-on-a-chip kind of microfluidic sorts of concepts that allow us to capture biomarkers and biochemical signatures of health status that can either be used separately or combined with electronics to provide a richer range of measurements that are related to health. And so, as was mentioned in the introduction, I've been here at Northwestern since 2016. That move motivated primarily by an interest in getting my group more coupled into a large, diverse, active, research-oriented medical community, which is what we have here in Chicago. So most of what we're doing, again, is at the boundary between engineering and medicine, trying to bring to bear new engineering principles to improve outcomes for patients, ultimately. And we do a lot of that work in the context of this institute that was mentioned. So the vision that we've had, and I think a large number of other groups by now are pursuing as well, is the idea of reformulating microsystems technologies, the engineering foundations of integrated circuit technology, optoelectronics, photonics, microfluidic systems, into platforms that are more compatible with biology from a standpoint of mechanics and geometry primarily, but with a deep consideration around materials compatibility as well. And sometimes it's easiest to think about that in the context of the human brain, which is biology's most sophisticated form of electronics. And if you want to understand how the brain operates, or you want to develop modes for interacting with the brain to improve, health outcomes for patients who are suffering from various brain disorders, you might want to bring to bear to that problem man's most sophisticated form of electronics, a silicon integrated circuit. But doing that is fairly challenging, because there's an extreme mismatch between the geometrical features of the brain, the mechanical properties of the neural tissues, and the silicon wafer. And you can imagine, that type of integration would be challenging unless you really reformulate the mechanics and the materials that are used in circuits to make them look more like the brain. Is the idea of softly laminating and following, the contours and the gyral surfaces of the brain in a way that you would allow for a persistent, stable interface to the brain. And so that's kind of what we're thinking about, but not just in the context of the brain, all kinds of other points of interfacing with the body as well: the heart, the kidneys, the spinal cord, the peripheral nerves, the lungs, the bladder, all sorts of organ systems that where we have active programs. And I'll focus today on the body's largest organ, which is the skin, and thinking about electronic interfaces, microfluidic interfaces to the skin that qualitatively go far beyond anything that's available in a traditional wrist-mounted wearable type of technology, to kind of recapitulate clinical grade measurements, but in platforms that are physically imperceptible, almost like temporary tattoos or band-aids is kind of what we're thinking about. But in that broader context, if you want to do biointegration, if you want to develop man-made microsystems technologies that can interface with various parts of the body, it's useful to kind of think about the design engineering approaches that you see in the human body. And it's really daunting to think about them as an engineer, because the sophistication is far beyond anything that's accessible with man-made technologies. I mean, these are 3D systems that are active at the level of fluids and ion transport, and they integrate hard and soft materials, across length scales that go all the way from sort of molecular dimensions to macro scale dimensions. They're mechanically active and responsive and reconfigurable and so on. And so I think the idea of mimicking those design features in a man-made system is probably beyond what's kind of feasible, right, in the near term or the envisioned future. But you might imagine abstracting certain key features that appear over and over again in biology, and think about those features and ways to embody those in engineered systems. And that's kind of the approach that we've taken. And we have kind of three streams of activities along those lines. The first of which will really form the core focus of this presentation, which is in the development of tissue-like, skin-like electronic and microfluidic systems. So devices that are designed with mechanical, thermal, mass characteristics very similar to those of the epidermis itself. So you only think of them as like a second skin, and you interface them with the body. They can remain in place, exploit that interface for precision measurements of underlying physiological processes, peel them off when you're kind of done, and use that data in ways that sort of advantage the cost or the efficacy of healthcare, moving beyond sort of clinical and laboratory settings into continuous monitoring in the home and during natural daily activities. So that's kind of one area, that's the focus. I just mentioned we have two other streams. They're somewhat overlapping, but also distinct in their orientation. The second one is in thinking about transient or bioresorbable electronics. So how do you put together electronic circuits that have as their unique defining feature an ability to dissolve in water to biocompatible end products, so that you could imagine engineering an electronic sensor system that could be implanted into the body, monitor a wound healing process, an internal wound, and then naturally dissolve and disappear after that functionality is no longer needed, after the wound healing cascade has reached an endpoint, to avoid what would otherwise be a secondary surgery for extraction. That would be another example. And then the third area is thinking about three-dimensionality, which is an essential feature of any living system, these kind of open architectures in 3 dimensions that you see over and over again in biology. How do you do that, in a man-made electronic system is another direction that represents a centroid of activity for us. But I'll just focus on this first one, just wanted to mention these other 2 as well. So from a mechanical engineering, a materials engineering standpoint, the real challenge is to think about how you go from this kind of technology to, again, something that looks a lot more like biology from the standpoint of geometry and mechanics and biocompatible constituent materials. And if you think about that as a challenge, you might naively conclude that a silicon wafer-based technology is never going to be biocompatible, and the only way to build biointegrated systems would be to develop entirely new classes of electronic materials, get rid of silicon, replace it with polymers or 2D materials or carbon nanotubes, whatever is your favorite exotic academic type material, and move down a path on that basis. And we certainly spent a lot of time thinking about those kinds of strategies. We still have some programs. The other way would be to view it as more of a challenge in mechanical engineering rather than material science, and ask the question, are there ways to deploy silicon in non-wafer based formats? Would there be sort of simple design features that would allow you to leverage all the sophistication that's already been built up around silicon integrated circuit technology over the last 50 years, but reformulate it and adapt it in ways that would allow these kind of biocompatible physical characteristics that I mentioned a couple of times now. And it turns out that pathway has been much more fruitful for us. And that's what I'm going to describe just at a high level in the next few slides. And then, as I mentioned before, leapfrog out into kind of system level work that we're doing now. And so the answer to how can you construct a silicon integrated circuit that's soft and flexible and stretchy and curvilinear like the tissues of the brain turns out to be really simple, from a mechanics standpoint. And I'm sure everybody in the audience will find these 2 ideas to be extremely simple, but it turns out they're very powerful also. And the first one is sort of what you would learn in freshman mechanical engineering course or physics course, solids mechanics, which is that the bending rigidity of the flexural stiffness is a function not only of the Young's modulus of material, but also the geometry, the thickness in particular. And so a silicon wafer is a rigid planar piece of material. It's brittle, you can't bend it. But if you make silicon very, very thin, it becomes very flexible and floppy due to the cubic dependence of the flexural rigidity on thickness. And so that's what we do. We developed ways to shave off the near surface of a wafer, very thin ribbons and membranes of silicon, thick enough to support electronic functionality, but thin enough to be bendable. And so that turns out to be a pretty powerful way to make flexible silicon circuits, because you can take those ribbons, you put them on a sheet of plastic, and then do all kinds of processing. You can build circuits that way. But that's not quite what you really need to wrap the brain or the heart or even integrate with the skin. You need something that goes beyond just bendability to stretchability, as defined by elastic mechanical responses to large strain deformations. And that requires a second idea, again, super simple, but very powerful as well. As you take those thin ribbons and you integrate them with an elastomer substrate, and you do that integration not in a planar geometry, but in a wavy, kind of buckled, rippled geometry. And now what you have from a material standpoint is a composite material, a hard, soft composite structure, but deterministically engineered in a way that allows for an end-to-end stretchability with a modulus characterized by the underlying elastomer, that allows for these kinds of large deformations without fracture in the silicon, because the wave geometries can change to accommodate the applied strain. So you stretch this thing out, the amplitudes of those wavy silicon structures go down, the wavelengths go up. The opposite happens when you compress the structure. The amplitudes go up, wavelengths go down, just like in accordion bellows. And that's basically it. I mean, it turns out the mechanics of that kind of structure are kind of non-trivial. You have to work through the details, but at a high level, that should be pretty simple to understand. Now, those ideas, as illustrated in this slide, is just in the context of the materials, but you can embed all kinds of functionality in that sort of framework. And you can exploit not only that out of plane buckling, but also in plane buckling. And you can do it not only in one dimension, but two dimensions. So now you start thinking about filamentary serpentine mesh type constructs that support all the brittle, active, high performance electronic materials, where the stretchability comes as a result of integration of those mesh structures onto an elastomer like a silicone polymer. And if you do that integration and you study the geometry, you think about the thickness and the curvature and the fill factor and so on, you can really engineer these platforms to replicate an effective modulus that can be quantitatively matched to a biological tissue of interest, the skin in this case. You end up with stress-strain responses that are completely aligned with the human epidermis. And so when you put this device on the skin, the interface stresses are very much minimized, and so there's really no physical perception of the presence of the device. The device can also follow all of the natural deformations, the bending and the stretching and the curvilinear textures of the skin quite nicely. And so this turns out to be a very generic but very versatile approach, because it applies to wide-ranging classes of materials, including all of the materials that are used in modern silicon integrated circuit technology. So that's basically it. Those are the ideas. It's mechanical engineering fundamentally, with some material science concepts mixed in there. And then you use those as starting points. You add all kinds of engineering elaborations onto those basic ideas, and then you can kind of do whatever you want, you know, in terms of skin-like electronics. And we felt like we kind of figured this out in 2011, got a little bit better in 2014. And at that point, begin to sort of ask the question, what is this good for? So now you can put electronics on the skin. Like, what do you do with it to improve patients' lives? You know, can you impact medicine in the way we think about human health, starting with these engineering concepts? And you know, in order to make all of this work, it becomes an exercise not only in electrical engineering. You always have to think about the circuit topology and the functionality at that level, but now you have to embed mechanical engineering and mechanical design quantitation in parallel with circuit design. And so we work closely with Yonggang Huang's group here at Northwestern. He's a computational mechanical engineer. And so we can do quantitative system level FEA modeling on these systems to get the layout such that you know, we can achieve ranges of stretchability that match requirements depending on body location and point of integration. So that's where mechanical engineering. So mechanical design now happens hand in hand with circuit design to get the kind of overall system level properties that you ultimately need. So that's from the standpoint of mechanics. What about interface topology? So you can obviously sort of accommodate large scale curvature, large radius of curvature kind of surfaces, but even micro topology, you have to worry about that, because you're thinking about these devices not only as intimately interfaced to the skin from a mechanical standpoint, but from a measurement standpoint as well. You'd like to minimize that interface impedance, and one way to do that is maximize the aerial contact. And so these filamentary serpentine mesh structures do a good job on the mechanics, but also in the geometry. And that's what's shown here. This is a colorized scanning electron micrograph of one of these mesh structures sitting on a polymer replica of human skin, which is extremely irregular and highly topographically complex. But these type of geometries do a very, very nice job as sort of accommodating that kind of surface texture. So this is another important feature of the devices. So now that you have all of that, you put the relevant electronics, the relevant biosensors, you can begin to recapitulate the kinds of measurements that are done routinely in a hospital with taped on sensors and wired connections to external boxes of electronics in these skin-like devices that can do that monitoring continuously outside of a hospital, not only in the hospital, but outside of it as well. So you can measure things like heart rate, which you can do with a wrist-worn wearable, but now you're talking about heart rate in the context of electrocardiogram. So you get the full waveform of electrical activity associated with each cardiac cycle. And that turns out to be much more powerful in terms of assessing cardiovascular health. And that really demands persistent contact with the skin. And I think in order to do that, you really need this kind of skin compatible mechanics. Otherwise, you end up with irritation at the skin interface. The other thing you can do that requires skin contact outside of sort of electrocardiograms would be sort of measurements of hydration level of the skin. A lot of skin disorders are associated with low hydration levels. It'd be nice to monitor that in terms of treating patients who suffer from those kinds of skin diseases. It's very easy to do that with these platforms. And you can also measure sort of time dynamic physical characteristics. So you can embed pressure sensors. You can measure sort of the time dynamics of pulsatile flow of blood through near surface arteries. And you can do all of these different things in a single platform, which turns out to be critically important. So that multimodal measurements of health status is ultimately what you really need. And sort of packing all of that functionality into a single platform is the direction of the future. So we've probably written, 50, 100 papers, all kinds of different biosensors, all kinds of different functionality that's compatible with that mechanical engineering concept that I stepped you through. And a lot of other groups are doing great work in this area as well. And so there's quite an extensive toolkit now of precision thermal measurements, electrical. I'll get back to sweat. You can embed microfluidic collection and analysis capabilities into these kinds of devices, various kinds of mechanical measurements, strain, motion, modulus, pressure, optical, mechanoacoustic, so you can capture body sounds, almost like a digital stethoscope is the way you can think about it. And I'll get back to that in a little bit. But anyway, a broad range of capabilities. But like ultimately, as an engineer, you would want to have some kind of practical impact ultimately. I think there's a lot of great academic science, and we try to do that as best we can, and a lot of our focus is there, but we're kind of looking beyond and asking, like, what is the consequence of all of this stuff is something that we're very interested in as well as engineers, biomedical engineers. Ultimately, that's what we like to do. So we started with this particular patient population as an area where we thought we could make a contribution. And it's, in the context of premature babies. So these are babies that have been, delivered far beyond, kind of term, time point, and their health status is very fragile. You go into a neonatal intensive care unit, this is what you'll see. It's tapes that are affixing biosensors to the surface of the skin, wires connecting those sensors to external boxes of data acquisition electronics, highly invasive to this patient population because the weight and the bulk and the mechanics of the wires frustrate natural movements. These adhesive tapes, application and removal on a 24-hour cycle can in many cases lead to skin injuries upon peel back of the tape because the skin is highly underdeveloped at this age of the baby's life. And also the presence of the wires just frustrates all kinds of routine aspects of clinical care, and it prevents parents from engaging with their child. The presence of the wires really restricts that as well. And so about four and a half years ago, we decided, if we could really make this wireless skin-like biosensor technology work well, then we'd like to bring it to bear on this set of clinical unmet needs to get rid of the rat's nest of wires, replace it with a few of these skin-like patches, and replicate this kind of functionality in a way that would be much more humane to the babies and also to the parents. So this is just Photoshop. I created this slide maybe 4.5 years ago, but that's kind of what we wanted to do. I won't go through the details. There's like a ton of engineering that needed to be done, but based on the concepts that I've already described, it turns out that you can recapitulate all of the vital signs monitoring functionality that's done in even a very sophisticated level 4 NICU in these wireless skin-like device platforms. It actually requires 2 devices. One shown here goes on the chest to measure cardiac activity. The other one goes on a limb, typically the hand or the foot, to measure blood oxygenation. And these devices operate in a battery-free, fully wireless modality, so we don't have the weight or the bulk or the safety risk associated with batteries, and we are able to achieve that full skin-like form factor. So here's the device that goes on the chest. This peanut-shaped serpentine structure, that's the antenna, and it's doing wireless data streaming. It's also doing energy harvesting. You put that on the chest, you can capture these electrocardiograms. I mentioned that before. All of the tests that we've done on premature babies have also involved simultaneous recordings with these wired clinical standards, so we can do quantitative assessments of data quality. These devices can operate at very high level, as you can see here. And from these data streams, you get heart rate. Heart rate variability is typically what the physicians are looking at, but you get the full PQRST waveform as well. From the depth of these peaks, the time variation of the depth, you can get the respiration rate. We also have a temperature sensor. We get the chest temperature. So that's a fair number of different vital signs that need to be tracked. But as I mentioned before, you also have to do blood oxygenation. That requires a separate device. And it's an optoelectronic rather than electronic interface to the body. It's a pair of LEDs, red and IR. They flash on and off out of sequence. We measure the backscattered light at an integrated photodetector, continuously wirelessly stream the response of that photodetector to an external computer, and that allows us to capture plethysmograms, and from that data you can determine blood oxygenation. So that's kind of how it works. You put it all together, this is what it looks like on a patient. So this is a 31-week gestational age premature baby in a NICU at Lurie Children's Hospital here in Chicago. You can see the size, very tiny. This hand here, that's Aaron Hombas. He's head of neonatology, a key collaborator here. You can see the device, you can see the wired-based systems, and we have an antenna in the base of the isolette that's delivering power to the device and also pulling data continuously back off of the device is the way it works. So we've done about 150 premature babies so far, no adverse events. Everything looks very good. And in fact, we've expanded beyond the neonatal intensive care unit to the pediatric intensive care unit. Same need for wireless operation, but the patients are older and bigger and a little bit less fragile. And this is an example of a baby in the PICU. You can see the device on the chest. There's the one on the foot engaging with his mom, in this case, without the wires getting in the way. In this case, we have the antenna in the base of the chair, sending data, power up and pulling data off of those two devices. They're time synchronized as well, which turns out to be very important. Babies are very happy. The mothers are happy. It's been a great project for us. You can see the kind of skin-like mechanics. Here's the baby rotating around. You see the wrinkles in the skin. The device follows those wrinkles without any mechanical constraint. In this case, we're picking up ECG from the back, and that works really well also. So I would say a project like this, you know, it's foundational engineering science, but to have an impact in medicine, you really have to engage with the clinical community. So the co-authors on this paper is published a little bit more than a year ago, include you know, dermatologists, neonatologists, folks who specialize in pediatric, nurses as well, turn out to be very, very important collaborators here also. So this is the kind of data, this is the most primitive representation of the data. From the ECG, you get heart rate. From the PPG, you get SpO2, blood oxygenation. This is a bland Altman plot. So this is difference in the heart rate measured with our device compared to clinical standard as a function of the mean of the heart rate measured using those two techniques. And so the tight distribution you see here is within medical standards, and this is 4 million data points. So you think about big data, this is it in a lot of ways. And this is just, again, the most basic level of interpretation of the data that you could imagine. There's a lot more information content associated with the full recordings, and we're looking at that. So once you have kind of a foothold, once you've reproduced what's done as part of clinical standard, now you can think about going beyond. What would a NICU of the future look like, and what kind of monitoring would be powerful? One thing that you know, the doctors and nurses would love to have access to is a continuous beat-to-beat measurement of blood pressure. Very difficult to do that with a cuff or an arterial line with a premature baby because the arteries are so tiny, the tissues are very easily bruised. And so you like to do that in some other way. And it turns out that the pulse wave velocity, that is the time delay between a contraction of the heart, a single cardiac cycle, and the time when that pulse of blood arrives at the hand or the foot, turns out to be a pretty good substitute for systolic blood pressure. And that's what you're seeing here. This is a PICU subject. We're measuring arterial line in the red and the pulse wave velocity or pulse arrival time measurement captured with this time synchronized pair of skin-like devices is shown in the blue. So there's some opportunities here now to use engineering innovation to go beyond and to provide additional data streams that are critical in the assessment of health status of these babies. And that represents one direction of our work. The other direction is try to take what we have and get it out into distribution where people can use this technology. And Bill Gates personally got interested in some of this work, and he's funding a large-scale deployment into lower and middle-income countries where there's no monitoring technology at all. Can you put wireless devices in that context, and can you drive the cost structure down to sufficient levels that would allow practical deployment in those parts of the world? And it turns out that all of that works, and I won't go through the details, but we're in the middle of a 10,000 unit deployment into India, Pakistan, Zambia, Kenya, and Ghana. I spent a couple of weeks in Zambia myself in December, and this is very, very exciting, at least for me and the students and our collaborators, right? Get our technology out into places where it can have a real impact. The engineering features of these deployed devices has to be quite a bit different than what we were deploying at Lurie Children's and what I just described to you, because the level of ruggedization needs to be much higher. The devices need to align with conventional manufacturing practice to get the cost down. We've added some additional sensor functionality and other things, and this was just published here in the spring. So this is kind of the newest work that we have in this space. It's currently deployed in 17 countries. I mentioned the LMICs, but we're deployed in other parts of the world as well with various partners. It's a great effort thus far. So that's kind of all I had to say about neonatal and pediatric health, but these same concepts can be deployed not only sort of beginning of life, where we're really focused on maternal, fetal, neonatal, pediatric, but also end of life. So stroke survivors, Alzheimer's patients, Parkinson's patients, that kind of thing. And this is the collection of devices using these same engineering principles, again, mechanical engineering at the core, for other kinds of measurements. So we have flow monitoring systems. We can do functional near-infrared spectroscopic measurements of cerebral hemodynamics. We can measure fetal ECG. I'll say a few words about this device, neonatal EEG, and different types of monitors for tracking surgical recovery in pediatric patients. So the fetal ECG is also deployed at scale through the gates. So this is 487 pregnant women, mainly in Zambia. So we're tracking them throughout the intrapartum period, and we can measure not only pulse arrival time, respiration rate, SpO2, and heart rate, but correlations between all of these different parameters. And so there's a lot of kind of discovery enablement that flows from these data streams that allow you to determine not only instantaneous health status, but hopefully some kind of predictive sense of the trajectory of where they're going in terms of their health at any given moment. So let me say additional words here about this particular device, which we originally developed to measure swallow and speech in stroke survivors who are undergoing rehab to relearn how to speak and relearn how to swallow. These are patients who suffer from something known as aphasia and dysphagia. And we're working with folks at Shirley Ryan Ability Lab here in Chicago to develop devices to monitor those kinds of characteristics. And because of the soft mechanics that we've developed in these devices, you can really put them anywhere on the body, certainly not constrained to the wrist, but even in very sensitive locations of the anatomy. And this is called the suprasternal notch. And it turns out you put a high bandwidth accelerometer right there, you can measure all kinds of physiological information. Swallowing and speech was our initial target, but you can measure lots of other things from a single device at that location.
What if you
don't want to have a microphone. The microphone has all kinds of types.
I usually just watch. I watch like college football.
That's what
the Packers.
Not
The Packers.
Very useful in a broader context. But here's what some of the data looks like. So this is a raw signal, an individual sitting in a chair quietly. This is what the data looks like. It looks like it's really noisy, but it's not. This fuzzy stuff, that's actually associated with cardiac sounds. So you apply low pass filter, you get the cardiac information. This is kind of the lub-dub. It's like the stethoscope functionality of the device. And you can determine heart rate and heart rate variability. You subtract out the cardiac, you're in the, left with just respiratory information. These oscillations, that's just chest wall cavity changes associated with respiration. And then these direct delta function-like features, that's swallows. So you can see the patient swallowing here, 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 and here. These patients have to relearn not only how frequently to swallow, but when to swallow relative to the respiratory cycle. And you can kind of think about this yourself. You actually want to swallow at the height of inspiration or at the depth of expiration. You don't want to try to swallow in the middle of that respiratory cycle or you'll choke yourself. And so, these stroke survivors have to relearn stuff like that. So this swallow is fine, that one's fine, that one's fine. This one's a little iffy, should have swallowed a little bit earlier. And so this kind of device can help to sort of, allow rehabilitation specialists to work with these patients to try to recover those kind of instinctual aspects of swallowing and respiratory activity that we take for granted. But anyway, you're close enough here at the suprasternal notch to the carotid artery. You also get, as I mentioned before, heart rate, you get swallow, you get speech. You also get other kinds of respiratory functions like coughing, sneezing. You get body dynamics and orientation and so on. And so a number of folks in the Chicago community were aware that we had this device. COVID hits in March, and they come to us and they ask, hey, can you adapt your stroke monitoring device to monitor the key symptoms of COVID? Because you can't monitor those symptoms from the wrist because they consist of fever, cough, and shortness of breath. You can't do any of that with a Fitbit. And so this device, we adapted it, aligned it to those requirements, those 3 key symptoms, and have deployed them at scale onto COVID patients. So we have full automated way to use these devices. They deploy onto the patient. They interact with a charging pad. Data gets wirelessly transmitted up to a HIPAA-compliant cloud. We have a graphical user interface, so physicians can look, for example, at coughing frequency as a function of time to track a patient's progression as they move through various stages of the disease, but also to monitor frontline healthcare workers so you could potentially pick up very early symptoms of the disease, pull those individuals out of a community to reduce spread. So that's kind of the idea, and we've deployed on about 250 subjects at this point, 20 weeks of deployment, 10,000 hours, about a terabyte of data. So this is ongoing work. This is an example of a patient. This was a nurse who contracted COVID as a result of dealing with patients at Northwestern Memorial Hospital. She only has 1 lung, so she was very, very high risk. And we gave her a device. She wore it throughout the period immediately after she was diagnosed and went into the hospital, and also continuously when she was released back into the home. So we're able to really track this patient and a number of other patients as they move through various stages of the disease. You can coughing here. The cough counts, that is the frequency of coughing per hour for this patient, reached levels of about 150. And then you can see that decay over time. Or measuring heart rate. We can see undiagnosed sleep apneas, transient tachycardia events in the heart rate, all kinds of key signatures of the disease that may help to understand the disease better, as well as being directly relevant to tracking and caring for individual patients. So with the last 3-4 minutes that I have, I'm going to talk about microfluidics, because everything that I've discussed so far involves biosensors that operate in the realm of sort of mechanical or thermal or electrical measurements. And that's great, but your human body is a biochemical factory in a sense. And if you really want to get deep insights into health status, you'd like to have biomarkers based on biochemical assessments of health, as well as these kind of physical-based measurements. And so we spent some time thinking about whether you could, come up with a classes of materials and design approaches that allow you to take conventional lab-on-a-chip type technologies, render them in skin-like forms, interface them with the surface of the skin, and then use them to capture microliter volumes of sweat as it emerges from the surface of the skin, move into a network of fluid channels with colorimetric or electrochemical assays to detect certain biochemical species and their concentrations over time. And we've been able to do that. There are a number of other groups working in this area of sweat analytics, but I think our platforms are uniquely defined by this idea of lab on a chip, lab on a skin, microfluidic type approach is a little bit different than others. But this is a kind of example. We have a Serpentine channel here. It's collecting sweat from an inlet port in the center. And we use this to measure sweat rate, sweat volume. And then we have 4 circular reservoirs that have colorimetric chemical reagents that respond through a color change to concentration of lactate, chloride, glucose, and pH that tends to work pretty well. Again, mechanical engineering, critically important here, because not only does it need to be skin compatible, as with the electronic devices I talked about previously, but it also has to have a watertight seal to the skin. It can't leak. And that turns out to be fairly challenging. So you have to use all kinds of modeling approaches to understand and optimize the mechanics, choose the appropriate materials, the layouts, and so on, to enable that kind of skin tight seal to the skin. But that all works. This is a device you can see on the outer form. The graduate student who's working on this, there's the fill front. You can just read that out visually. You know the geometry of this channel, the size of that inlet. You can determine the amount of sweat that's been released from that local area of the skin. It turns out that calibrates to full body sweat loss. And so you can measure total sweat loss by just eyeballing that channel, and then color change in the other reservoirs allows you to get additional insights. But here's kind of what it looks like, different channel geometry without the colorimetric reservoirs, just to give you a sense of how this all plays out. So this is a design, kind of a serpentine, structure. This is on my forearm while I'm working out in my basement. And so here we have a colored food dye at the inlet. And so as sweat moves into this device, you can just see it filling these channels. And it becomes an interesting kind of feedback on your workout. So about 6:19 or so, I got really ambitious and decided I was going to try to fill this whole thing all the way to the end. Almost killed myself. But by 6:35, I had filled the whole thing, which is a fair amount of sweat coming out of a small region of my skin. But at the same time, if you look at the surrounding skin, it's totally dry because the sweat is constantly evaporating. So you lose track of how much sweat loss you've suffered at any given point in an exercise event in the absence of a device of this type. And in this particular case, if you look at the cumulative sweat loss determined from analysis of these images, you can see that it depends almost perfectly linear on exercise duration. So once I got going, my sweat glands were operating almost perfectly at a sort of constant volume per time, in that sense, which is really, really amazing in a lot of ways. I don't think that type of thing has ever been measured before, but just interesting to kind of see that. So the other aspect of the development effort here is around these color changing chemistries and how to develop those chemistries and tailor them to physiologically relevant ranges of concentration that you would see in sweat. And we've been able to do that. I'm not going to go through the details. I'm almost out of time here, but this is the kind of color change you see for chloride concentrations that span the range that you would see in typical individuals. So some people sweat with very low electrolyte concentrations, maybe 10 millimolar of chloride. Others are at the very high end, maybe 100, 150, kind of up in that realm. But very vibrant, easily measurable, easily quantitated changes in color allow you to get chloride concentration. Same thing with glucose, a little bit more complicated. The chemistry here, lactate, I mentioned that before, pH, all this stuff works nicely. And then you can begin to embed more sophisticated concepts in microfluidics. Again, taking a look at what the lab on a chip community has done over the last years, very clever stuff, and just adapting it and putting it on skin, essentially what we're doing. So in this case, we have capillary bursting valves that allow us to sort of route the sweat around a platform that allows us to get some temporal information as well. So we have the burst valves here, and they cause a sequential filling of these reservoirs as sweat begins to emerge into the device through this inlet port. So by looking at, say, concentration of chloride measured in each of these reservoirs, we can see if it's changing during the course of a competition or an exercise event. Same thing with glucose and lactate. And then we get sweat rate over here. So that's kind of what it looks like. So after we published our first paper in 2016, Gatorade approached us. As you might imagine, they're very interested in providing additional data and information to their customers around how much Gatorade they need to drink and what type of Gatorade matches their body chemistry. So we put this together with Gatorade in the middle of launching. So it's two inlets, one that's measuring chloride, the other one that's measuring total sweat loss. And the idea is you give these out to athletes, they put them on and they determine, when they need to drink Gatorade. So this has been launched. There it is on Serena Williams. It's been on Paul George. It was unveiled in a commercial campaign that started the NBA All-Star Game in February. This has been a great engagement and a way to get devices out of the lab. And I'll just conclude with this. So you're excited as engineers to build things and you'll provide devices that might have value to people, medical primarily, but sports performance as well. But the whole world is going virtual as well, in a sense. And so I don't know if there are any gamers in the audience today, but the NBA 2K21 version of the game will allow you to buy virtual sweat patches. You can see it right there. In the game, you can buy these devices, put them on your character, and then use them to determine how to optimize a hydration strategy for your player. And so this is cool, but it's also a little disappointing because it means you can trivially add any function you want with software, and so as an engineer, it's a little distressing to see everything go virtual, but that's the world we're living in, and the software guys will take over and add functionality into the game. So with that, I'll go ahead and conclude. I think I'm out of time. I talked to you about skin-like electronics, skin-like microfluidic systems, and kind of what we're trying to do in areas of medicine and sports performance. All of this is very highly collaborative. So, I'll highlight Youngong Huang. I mentioned him a couple of times in terms of modeling, engineering science, clinical medicine, and sports. But the most important people are the students. Great group of grad students and postdocs, MD students, undergrads as well. All the hands raised here, those are undergrads. And I always like to conclude by acknowledging their hard efforts, great ideas. I just talk about this stuff, they actually do it. And I'll thank you for your attention. I'd be happy to answer questions if you have any.
Wonderful, John. Thank you so much for the phenomenal talk and the work. And I will start to read out a couple questions. The first question from Amadi, from our student, is related to the adhesion of the devices and our skin and tissue. Can you comment on that?
So I don't know if I caught the first part of that question. Is it the adhesion of the devices to the skin? Is that the question?
That's right, and also related to removal of the device.
Yeah. So so the devices for the NICU, they're really being used in a 24-hour cycle, roughly, because the babies need to be bathed. They need to have their diaper changed. There are other kinds of routine operations in clinical care that are happening roughly on that type of cycle. They could be worn longer than that. The power supply is either wireless or an onboard battery, depending on the device embodiment. But I guess ultimately the residence time on the skin is limited not so much by the device, the mechanical engineering or the material science of the device, but more by the biology of the skin. So your skin is constantly exfoliating dead cells from the very surface layers of the skin, known as the stratum corneum. And these cells differentiate and propagate from the base layers of the skin to that stratum corneum layer over a time scale of about 1 to 2 weeks. And so if your device is on the skin for longer than that time scale, you begin to accumulate dead cells at the interface between the device and the living skin underneath. And so it becomes sort of an unstable mechanical interface. The impedances tend to increase because you're just then measuring kind of dead cells. And so that type of 2-week time scale is being set by biology, not so much by the devices. So that's kind of how you can think about how long these devices can remain on the skin. It's not limited by the adhesive strength or the materials choices or the softness of the mechanics. It's really that accumulation of dead cells.
Thank you, John. Next question is from Mohammed. He is asking a power source. Are you using batteries, capacitors, or other instruments?
Yeah, so we have several different device platforms. The ones that I described initially in the context of the NICU, those were all wirelessly powered using magnetic inductive coupling, kind of RFID type technology, NFC type technology, same technology used to wirelessly charge your phone, but at a larger scale. So that antenna is in the base of the isolate, it's delivering power. That same magnetic inductive link is used for data transmission. For the devices that we deployed in Africa, that wasn't going to work because it required additional electronics and it required wall plug power to operate the antenna. And none of these places have a reliable source of electrical power. So we had to bring the power source onto the device in the form of a small rechargeable lithium polymer battery. And so that's what we did there. The devices for swallowing, that suprasternal notch device, it's in adults, it's fine, you use a battery. And the battery's a little bit more versatile because you don't have to stay within about 1 meter of that transmission antenna. It just, allows for a greater versatility in how the devices are used. So those are the two approaches. For the wirelessly powered devices, sometimes we'll include supercapacitors so we can have a little bit of onboard power in the event that the orientation of the antennas, the relative orientations change as a result of change in body position, transient change in body position. So sometimes supercaps, but they're continuously being charged and then they only get drained when the power transfer is interrupted.
Thank you. Maybe I can ask a question from myself. John, you mentioned you're delivering 10,000 devices under the Bill Gates Foundation, the program. how do you manage in your lab to do the scale-up manufacturing from a few devices to 10,000 and also ensure consistency?
Yeah, that's a great question. So we don't even try it because it's probably impossible. We do have some limited amount of manufacturing capacity here in the academic side of things. So we can do tens of devices per week, but at those kinds of scales, it's not really feasible. So we set up, in response to the Gates interest in deploying these devices, a very small kind of lean engineering oriented company. It's almost like a nonprofit, I guess, is the way you could think about it. But it's a team of, a dozen engineers and, kids who want to do something exciting. And they've partnered with sort of manufacturing companies here domestically in the US to outsource a fair amount of it. They do mostly quality control and final assembly and packaging in-house, and the electronics assembly, the flex board, manufacturing is done outside. And so it's kind of a bit of an outsourced model, but not entirely. They do sort of the final steps in manufacturing internally.
Very challenging task, but interesting. So next two questions from students. One, I'll make it short. One is about optimization of a location for placing a device, for example, on your arm. The other is related to disposable aspect. The student is asking, do you recycle some of the parts after using a device, or is totally disposable?
Yeah, so the first question has to do with positioning on the body. It really depends on what you want to measure, and you choose the anatomical location based on the measurement requirements. So you want to measure swallow, you put it on the throat. If you want to measure cardiac activity, you put it on the chest or the back. Kind of coincident with the chest cavity and where the heart is located. You want to measure digestive sounds, you put it there. You want to measure cerebral activity, you kind of put it on the forehead. So I don't think in any of these cases a precise positioning is required. In most cases, you have kind of a tolerance of maybe 1 centimeter in any direction. So the suprasternal notch is probably the location that's most sensitive to details of positioning. But even here, if you move it up a little bit, you move it down, you move it to the side, it'll change the relative amplitudes of different body processes that you're monitoring. Say, the respiratory amplitude compared to the cardiac amplitude may change a little bit, but it doesn't change the characteristic features associated with those various events. So you can use the same machine learning, digital filtering algorithms to separate things out. So positioning is important. It's typically not critical. You don't have to get back to exactly the same spot every time. There's a little bit of tolerance there. Maybe a couple of examples where that's not the case, but everything I talked about, certainly that's the case. The other question was around reusability and disposability. And I think it really depends on the use case. So here at Lurie Children's Hospital, a very sophisticated hospital, everything that they currently use that interfaces with these neonates is single one-time use, disposable. So all of the electrodes for measuring ECG, they just use them once, they throw them away. Even the wired NIRS devices they use for cerebral hemodynamics, they're disposable. And the cost is, for the ECG leads and the wires, it's maybe $20 per lead. For the NIRS, it's almost 120, but that turns out to be like a small cost component of the entire cost of having the baby in the NICU. So they just eat the cost associated with the devices rather than try to sterilize them and reuse them. And so that's our vision, at least for that type of environment. Devices would be single use. And you can run the numbers. It's probably, feasible to think about doing things in that way. On the other hand, in Zambia, Kenya, Ghana, these types of places, it's totally impossible. You cannot get away with that kind of mode of use. You really have to be able to reuse the devices. So when we ran cost models for the Gates, because they're not going to spend money and invest on anything that doesn't have a potential for a cost structure that would be compatible with that part of the globe, we ran numbers in terms of the cost per patient day. And in those kinds of cost estimates, if you project out in terms of volumes, you have to make some assumptions and so on. You're kind of in the range of a few cents per patient day. and that was deemed to be acceptable, for that type of use.
Thank you, John. We have many questions. So Laura, do you want to ask the student who raised his or her hand?
Yes, I will enable them to talk right now. I should be able to speak.
Can you speak up to ask the question?
Laura, it's my turn?
Yes, you may go ahead.
Hi. Hi, John. This is Xin Yu. Very thanks for the great talk, and it's amazing to see many wearable devices from your lab has been translated to the user side. So I'm curious about the practical use of those devices for long-term use, especially the attachment. Do you have any special treatment about the interface to make sure a user will not get device detached if there is, I mean, for a day or a few days of usage? Yeah, about attachment.
Yeah, I think there are a lot of opportunities. So our approaches have been fairly simple, I would say, in terms of the adhesive mechanics and the choices of adhesive materials. I think there's probably a number of things that one could think about that are much more sophisticated, maybe much more valuable also than the approaches that we're using. But what we're doing is just adapting FDA approved biomedical bandage materials and deploying them in kind of a double-sided adhesive. So they're sticking to the device on one side and then the skin on the other side. We choose chemistry on the device side. It's a little bit different than the skin side. So we have very, very strong adhesion to the device, but you want sufficient but not excessive adhesion to the skin, especially for these premature babies. As I mentioned before, the skin is, it's almost like a paste. It's like an open wound. I mean, it's not really skin. It's very fragile. And so there you have to pay a lot of attention to the nature of the skin adhesive material interface, what's going on there and how strong it is. And so you want it to be as weak as possible, but sufficient to keep it on robustly. And that turned out to be tricky. I mean, we spent a lot of time on that. One way, and we had, pediatric dermatologists were engaged in the effort, because that's probably the highest risk associated with these devices is damage to the skin on peel back. That's what they see with the regular tapes they're using for water-based devices. So it's not easy. I think it's a little more straightforward for us because we don't have a wire coming off, and so there are no forces being imparted to the device, like trying to tug on it to pull it off. So we don't have to worry about that. The other thing is that the mechanics involve a very low effective modulus. We have very thin devices, and so the interface stresses that develop during natural deformations of the skin are minimized. So they're very, very small. And so we don't need a very strong adhesive, turn out to be very important. But we're using like hydrogel materials that offer enough adhesion. They also are ionically conductive. I would say in terms of adhesives on the other end of the spectrum, probably the sweat microfluidic devices require the strongest adhesion because, as I mentioned, it has to be watertight to seal. Otherwise, you can't accurately measure sweat loss, sweat rate. And then you got athletes, running around and banging into one another and all kinds of forces, being imparted. We had devices deployed on triathletes in Hawaii. They're swimming in the ocean, they're running, they're biking. And there you really have to dial up the adhesive strength quite considerably. But still, at a level that's not going to, cause pain, when you peel the device back. But there you're talking about adults. It's much easier from a patient perspective, but that's what we've done. I mean, I think what you'd really want is some kind of, and I know a lot of people work, especially in mechanical engineering, on different kinds of adhesive approaches like structural, features and adhesives and so on. Something that you could switch, it would be adhere very, very strongly, and then you flip a switch and it would just fall off, and I think there have been, various studies along those lines, and maybe directionally dependent heel strength. I think it's kind of a rich area to think about. Like I said, we've just taken really simple approaches, but there are a lot of people out there with much more sophisticated ideas on how you engineer interface adhesion. And I think a lot of those ideas could be very valuable in these sorts of contexts.
Thank you. And it's also amazing to see you show us the FEM simulation and mechanical analysis. This is something, some aspects you usually don't show in other talks. That's amazing to see those ones and the mechanical designs in those devices. All right, thank you, John.
Thank you, yeah.
John, we're running out of time, but here are two comments made by the audience, and you might have thought of them. One is, have you ever thought about applying the flexible devices, for example, pressure sensor in wind tunnel beyond our body? And the other is the monitoring of wound healing status while using these devices. I guess you have thought about them.
A little bit, yeah. Structural health monitoring, I guess, is maybe a more general way to think about the comment on the wind tunnel. So living airframes and aerospace, different things like that. I think it's really interesting direction. We focused on human health a little bit more, but yeah, I think it's a reasonable, comment. And yeah, what was the other one?
Wound healing.
Wound healing, yeah. So we've done a little bit in wound healing. The problem is, you have to get IRB approval to use devices on human subjects. And if it's totally non-invasive, it's not an issue. Once you put something directly on a wound bed, it's very hard to get approval for that. That's kind of a practical thing. The other challenge with wounds is they're very heterogeneous. They're messy. I mean, they're wounds. And so I think you have to think about dense sensor arrays, and it's definitely a direction that we want to go, but I don't have a lot of examples. Maybe we publish a couple papers, but I think it's important. It's a great suggestion. There's some challenges there, but maybe opportunities as well.
Thank you. So maybe last question, John, also from a student, and I'll paraphrase it because it's also my question. I'll make it grander scale. So if you look at your lab, both at UFI and Northwestern, you were able to publish some of the best papers in some of the best journals. In the meanwhile, you're doing quite application-driven, impactful translational work. And to many of us, it's a fight between them because mostly the articles in Science and Nature are more on the basic side. And for the applied journals, they're usually more on the engineering side, but you are covering both at the same time. And can you share your experience with us and how you manage to do both?
You gotta have a very high attempt frequency. Maybe that's, you have good statistics, I would say I don't have any like formula or specific strategy. I feel like, if you have an idea, it's a little bit different than things that people have published in the past. That would be one thing that's important. If you're doing sort of engineering science, I think for those particular types of journals, it has to be something that looks different to people, especially the editors. They look at the figures, say, oh, I've never seen anything like that before. Roughly, you need to be kind of in that realm. If you can add on top of that some kind of capability that wasn't available previously. So you had kind of an idea and then a consequence of that idea clearly demonstrated. Those are two features that are probably really important for those kinds of journals. Not that that should really be the aspiration. I think a lot of academics, and my students and, sometimes myself as well, get a little bit too wrapped up in that. I think you want to develop a portfolio of publications that are impactful, rather than like, I got my science paper and that's great. It's like I think it's better to judge a researcher by the net accumulation, right, of their work, and that might appear in a sequence of papers, right? So that's just another thing. But some level of novelty around the engineering or the materials or the device geometry or the measurement modality, and then some kind of unique consequence of that. The other thing that's been helpful for us, and this has just been my style, I guess a lot of other people probably have this style too, but I got my career started at Bell Laboratories, and I think interdisciplinary collaborative efforts, you know, can lead to much higher levels of impact and significance. So it's not, you know, electrical engineer sitting around and building a circuit and speculating about how it might address a clinical need. You're in the trenches working with the nurses, right, and deploying devices on patients, you know, in the context of what I'm talking about. I think that could add a lot of technical depth and broader relevance, broader impacts, right? And so if you can put those three things together, if you can check those three boxes, interdisciplinary, broader impact, novel capabilities, some new kind of device concept, then I think those are kind of the key ingredients, I would say, for an engineering-oriented paper in those types of journals. I mean, the other way to do it is focus on new science, right? And that's kind of obvious. But if you want to try to get outside of the more traditional academic scientific focus, and you want to strictly define, and you want to publish in those journals, then those are the three key pieces, and you want to do the best you can in each of those three. You don't have to have every one of them, but the more that you have, probably the probabilities are going up a little bit. But I would say it's random, you know. At the end of the day, I can't figure it out. Sometimes I submit something, I think it's like super awesome, and then it doesn't go anywhere. Other times I think, it's pretty good, we'll see what happens. And then it goes through. So I don't know, it's random.
Thanks so much, Sean, for the very candid, insightful perspectives. I intentionally asked you that question for our students to understand that is not really basic discovery, that's the only type of work that people care about in those journals. As long as we're solving a real problem that is truly impactful. For example, John's work, it's a super example that you can certainly publish in any type of journal if you really impact the world and solve a problem that people care about. So that's my intention of asking the question. I think what you said will be very, very insightful to many of our students and also faculty members. So we're running out of time, but to all our audience, I want to assure you that as long as the pandemic situation is over, John already committed to be a plenary speaker on our campus for an international conference I'm chairing next July. So you will have an opportunity to see John in person, to interrogate him with more questions if you have more. And I look forward to having him on our campus with all of us to inspire us further. But John, on behalf of everybody, it's already 3:20. On behalf of everybody, I thank you for the great talk, but more importantly, for being an inspiring researcher. And I look forward to seeing you on campus next year.
Great. Thank you for the invitation. Thank you for the kind comments. I'll see you guys later.
Thank you, John. Bye-bye.
Bye-bye.